you have this problem if you want to, without understanding the effects of changing rainfall patterns, which makes your
Hi.
job a lot harder. And I just want to add one other thing is that when you get into that, the effects of rainfall are not always straightforward, that they're mediated by hydrology. And so, for example, you can get really big malaria epidemics in Botswana in a place where it's never rained.
Yep.
And that sort of temperature, rainfall, hydrology relationship means that you're probably not going to be able to make simple predictions about the effects of, and the fact that we have organisms with real ecology means that you're not going to be able to get very, generalizable predictions, except for from temperature. But once again, the literature suggested that 50% or a doubling from temperature, but that's, in the scheme of malaria transmission, that's pretty small effect size. So I'll go off and let myself get skewered by everyone after saying that.
Hi. Is there any chance that I could talk?
Hey, Madeline, good morning.
Hello there. I am so sorry for
nice to
being
have
late
you back.
on this.
No problem.
Yeah.
We're going to call on you shortly, so just
Okay.
stay on the line. Before we go,
Okay.
because we're going to ask Dave a question here, but actually we're going to hold that and bring back. I really would like to hear from Leah. Is she there?
I am, and I can speak to some of these things about how we can potentially understand some of the links between multiple of these climate drivers, because I think that I think this is one of the things that we've always admitted when we've done our kind of temperature focused studies. Yeah. This is looking at one constraint, and the way that we can study this is based off of the data that we have that are actually available. I'm sure that many of you are experimentalists here, and you know how difficult these types of studies are to do to understand, like the inherent biology of these organisms. And there are folks like we are working with Courtney Murdoch at Cornell, who's doing some really nice experimental work manipulating both temperature and humidity together, so we can explicitly understand.
We've got some material prepared on that actually.
So there's that bit. I know there's one of my colleagues from here at Virginia Tech, Chloe Lahandra, is studying kind of behavior and also desiccation, and we're hoping to be able to do some evolutionary studies and understand that. But it's always really difficult when we have these types of correlated drivers for to understand what's going on. Like if we use our correlative approaches, the things that we all like to do that are regressions, or even most of the machine learning models, where you're trying to say, what is the suite of predictors and how does that relate to where we see malaria on the ground? Anytime that your predictors are correlated, your interpretation of the relative impacts of these different effects is really kind of hidden, right? Being able to tease out can often be very kind of fraught, actually. And in particular, then trying to use this to predict into the future becomes even more difficult, because the underlying relationship between those environmental factors is likely to change as climate changes. And so we're not really even able then to use those correlative models to predict into the future. And so
the species diversity is quite vast as well. So.
Yeah, I mean, we're not even talking about, the fact that these little bugs can evolve, right? So there's that whole aspect of it too. So of course we're going to need to simplify things in order to try to kind of tease out these different bits. And it also gets down to things like, and I'm sure that Sadie would be keen to talk about this, kind of your spatial scale, because your patterns with where, for example, in a particular locality, you might have substantially more variation in rainfall and humidity than you do in temperature. So of course, you're not going to be able to infer a relationship with temperature because you don't have enough breadth. So like what types of patterns we can understand actually depends at the scale at which we look for those patterns too. So I think that there's ways that we can use the types of information that we get from kind of larger scale studies and local studies and from laboratory studies together.
To put that together.
But we're going to try to put them together. I think that how to do this well is still an interesting and open question. But I think that we're all trying to progress this forward, and all of us recognize that this is not a simple.
I have to say it's quite a surprise actually, because I mean, if you read reports by Simon Hay from 2000, in the late 90s, there was a group of people who were very interested in climate, and then they stopped for some reason. But almost all of them mentioned this problem of the data. And we still have that same problem. And we're going to ask a few questions about that as well. So if that's okay, we can just move forward. Go ahead.
I mean, and I will comment that part of these things are just about the nature of
we have two people with their hands raised. Maybe they want to ask specific questions.
We can create some space afterwards a little bit, in a short way. Go ahead, Claire.
So one of the reasons why it's still like coming up as a problem, that because that whether we like it or not, this is just how like understanding data with statistics works, right? There are certain types of things that are underlying statistical issues about how we can parse out information and data and parse out signal from noise and deal with correlated predictors that don't go away, and they will never go away. At least I don't believe they will ever go away. So of course those problems are still there, and we're just trying to get creative about how we work around those problems. And I think we are getting more creative, but of course we're still probably going to be trying to address these problems forever.
Yeah, we've seen some incredibly interesting work, including especially on the thermal biology of mosquitoes, but also just figuring out the interaction with the pathogen as well. And you have the pathogen, the mosquito, and then you have the climate. Each one of them has different limits, and that can get complicated quite fast. And like Dave loves to remind me, there is also humidity and rainfall. So, Dave, this is your equation. Well, maybe not your equation, but this is from a book chapter you wrote with Richard Fisham a few years ago, measuring malaria transmission. And the question we have for you here is just to explain to our audience how climate change or climate elements impact these processes. This is an equation of malaria transmission for those who, but Dave is going to explain. Go ahead.
So very quickly, this is not my formula. This is McDonald's formula, published in 1952 for the first time, describing the reproductive number for malaria. So it's the number of malaria cases per malaria case. So it's describing how much parasite transmission gets amplified over a single parasite generation. So the first thing to say about this formula is it looks really complicated, and it shouldn't be. McDonald was actually not very good at math. And so in the denominator, you're seeing this scary figure, negative ln of p. What he actually did is he had an equation that was written in continuous time, and then he translated that into an expression for the probability of surviving one day. So if you actually take 1 over the natural log of p in that weird way of deriving it, it ends up being the expected lifespan of a mosquito. How long does a mosquito live? And if you actually write this in a slightly different way, then it makes a lot more sense. So if you take away all of the parts that have to do with humans and you just look at the mosquitoes, the things that are purely to do with mosquitoes, it turns out to come down to 3 terms. So the first term is how many mosquitoes are emerging from aquatic habitats. So something to do with, yeah, here we are. So some lambda is something to do with aquatic, the ecology. And then this FQ over G is the number of human blood meals per mosquito over its lifetime, which appears twice. So you have to get bitten, or you have to become infected and then survive through the extrinsic incubation period and transmit. So, 2 blood meals is FQ over G twice. And you get this expression at the top, which is mathematically, under the assumptions of McDonald's model, it's the equivalent to what he wrote down. And what I think is much more, why this is a useful thing is because you actually see what vectorial capacity means. So it tells the story of transmission. I emerged, I blood fed on a human and became infected. I survived through the extrinsic incubation period, and then I bit more humans. And so that expression is kind of, when you take it apart, one of the things that McDonald did, he focused just on the sporozoite part, and he focused on sensitivity to mosquito mortality. But if you put the rest of it in, the human biting rate part back in, and start adding things up, you find that some of these parameters appear more than once, and you can sort of make an ordering based on the number of times that they appear. So this is the second yellow column, where you just counting, you sort of, what's the order? And this is giving you some measure of sensitivity to parameters. But then there's this other part that ecology has to have something to do with adult mosquito populations. So if you put that back in, you get an argument that you change the order because eggs laid by adults is proportional to the same thing as blood feeding per lifetime. And so it's sort of F over G, sort of the number of blood meals that get, you feed and it gets transformed to eggs, and those eggs get laid. You get this sort of strict order of sensitivity. So the least sensitive would be lambda, the emergence rate from aquatic habitats. And then there's what fraction of blood meals are taken on a human, then the blood feeding rate 3 times, mosquito mortality 4 times, and the duration of the extrinsic incubation period, which has to do with the parasites, because they're also ectothermic while they're in mosquitoes, tau, that affects it once. And then you can sort of back that out and say, how is temperature and humidity and rainfall affecting all of these things? And you see that every single one of these things, in some way or other, and I put the strong effects, if you were to put the complete model of just everything would be connected to everything, because you can make plausible stories. But the strong effects would be relative humidity and temperature affecting almost all of these parameters, okay? And so this is just a way of sort of sorting out and thinking about how temperature and humidity would affect the parameters that affect transmission. And that's what McDonald's R naught expression, I think, is most good for in this context. Is that what you were looking for there?
Yes, thank you so much. Thank you so much, Dave. It's really the processes that lead to the parasite being transmitted from one person to another, and as mediated by mosquitoes, and therefore how the climate elements influence that entire process, starting with the emergence of the mosquitoes themselves. And I know that McDonald was very keen on the idea that the mosquito survival is the more important component of this equation. I think you hold the view that the numbers emerging are probably either more important or equally important. And so this rainfall thing is quite a big deal in this equation, right?
Yeah, I mean, I think the mistake is in looking at that formula and counting up the number of times the term appears, and therefore concluding that because it's sensitive to that factor in multiple ways, that the effect size will be big. And the classic one here is to really think sensibly about mosquito ecology and realize that mosquito populations can grow very quickly in a very short period of time. And that variability in lambda is probably what explains most of the variation in malaria transmission across the range. It's not actually, it's not sensitive to that parameter, but it's the most variable of all of them in the expressions that we look at.
No, thank you so much. Let's move forward. I would like to bring in Erin and Sadia here, if that's okay. This is a colleague of ours who no longer works on climate, who maybe still does, but did a lot of work on climate when it was still not a very attractive field. So this is Dr. Nabi Bayo's PhD thesis, and I would encourage all the young people who want to get into this field to really spend time reading that PhD thesis. He did this with Professor Steve Lindsay, and he did a lot of this work in the lab and generated some really, really good data. Erin, in the beginning, you were telling us about the unimodal nature of these equations. And Dr. Nabi had much earlier on suggested that, whereas models assume that this line is going to continue, in the field, in the lab, you actually see that if temperatures go beyond a certain point, the mosquitoes just die. So you have that unimodal nature nature of things. Could you explain just a little bit more what advancements have happened since then? And we have some slides later, we will talk about those, but it would be nice to hear how these things play, because you guys have really worked on this in much more detail since this time was this collection was done.
Yeah, thank you for showing these data. These are really, really, really important data that inspired a lot of our thinking in trying to figure out the thermal biology of malaria. Like you said, early on, it wasn't that attractive of a topic for people to work on. And then, around the turn of 2000, it really became popular because of the issue of climate change, as people started thinking about impacts of climate change on malaria, and the rationale being, malaria is a warm associated disease, therefore warmer temperatures are going to expand malaria. And what this data shows is the importance of that nonlinearity that I talked about earlier. So this is just the development rate between temperature and the Anopheles gambii mosquito at its immature stages. And if you use a linear model like this dashed one here, you can see that, the warmer it gets, the faster that development gets. But if you actually measure what happens to mosquitoes in the lab, you see that that development really drops off when you get to a temperature that's too warm.
Yeah.
And, Dave did a beautiful job explaining all the different components of the mosquito life cycle and how they fit into a model of transmission, and how those components have differential effects on transmission, in the sense that some of them have much more impact on the transmission equation than others. Like he mentioned, that adult survival being really important because it determines how many eggs they lay, how many times they bite, how long they live, whether they're able to live long enough to go through the incubation period, and so on. And so the fact that these traits are nonlinear is very important, because if you're just assuming that they all increase or decrease monotonically with temperature, it turns out if you build a model like that, you get a really different estimate of the thermal optimum for transmission. And what we found when we built a model using these kind of empirically determined nonlinear thermal performance curves for all of those traits that Dave mentioned, is that that's what lowers our estimated thermal optimum to 25 degrees Celsius, whereas the previous models that use these linear responses predicted optima around 31 to 32 degrees Celsius. So when you think about what is the perceived importance of temperature in the field for driving malaria transmission? If the optimum is 32 degrees Celsius, then in most places you'd be looking for an increase. So, warming temperature should mean more malaria transmission, because 32 is really warm. 25 is not nearly as warm. So in that range, you're actually not looking for a very steep relationship if you're already in a pretty tropical endemic place. And so that implies that where we should be looking for those strong effects of temperature should be places that have temperatures on the cooler end. As Leah was mentioning, how much variation actually is there in these different climate variables? And I think others have mentioned as well that these climate variables can be correlated. So it can be difficult to just use field data on climate and malaria transmission to infer these relationships. And these are the kinds of data that you can use from the lab, where you can directly measure how the mosquitoes and the parasites respond to temperature under controlled conditions.
So hold that thought, just for clarification. So if the optimum temperature for malaria mosquitoes was high, let's say 31, as previously thought, you are arguing that warming up the temperatures from an ambient of 25 would actually take us towards that optimum, correct?
Mm-hmm.
And that means that a warming climate would actually end up with more malaria. Instead, on the other hand, you're saying that the correct optimum is much lower than that, around 25, 26. I think we have some slides on that. And therefore, if the climate warms even by 1 or 2 or 3 degrees up, it goes towards the dangerous end. And that then is the basis for the argument that climate change might actually be an accelerant for malaria elimination. Is that a good way to put the problem?
I'm not sure I'd like to go that far because of all the other factors that you've been talking about, and everyone on this panel has been talking about, drug resistance and invasion of new vectors and so on.
Yeah.
But if anything, climate warming is not going to increase malaria transmission when you're already near that 25-degree range. That, climate warming, again, kind of echoing some of the points that others have made, climate warming is not necessarily the biggest threat when you're already in a pretty suitable environment for malaria. Climate warming is certainly an important threat when you're at one of those kind of marginal locations where, you know, that's why the highlands have been such an important case study, you know, because it's very clear to see malaria moving uphill in response to warming temperatures.
Okay, hold that. Wait, let's sorry for interrupting that, but I think it's a much more interesting section. I'm asking my colleagues to kindly mute everybody who is choosing to unmute themselves. So if you don't mind, I'm going to request Idris to hold that thought a little bit. I would like us to go back to a more health system part here. Also an important aspect, and I would like to invite Madeleine, if that's okay. There's only one question we have here, actually. So this is an operational framework for building climate resilient health systems. The previous iteration didn't have the low carbon component. The current iteration has the low carbon component on it. And the argument is that, you know, our health systems need to be both climate resilient and also carbon neutral. Some of these health systems are in fairly low income settings. They have a lot of other things to deal with. What are your thoughts on how countries dealing with diseases like malaria, dengue, and so on, low income countries, should interpret this kind of frameworks? And how actually do you achieve a carbon neutral health facility in my village?
Hi, can you hear me?
I can hear you very well.
Okay, so I sort of feel like, oh, I could have got into the previous discussion because that's so up my street. But this is also a very interesting space. And for those of you that have been following or engaged at COP28, you'll be very aware that there was a health day for the first time ever, which is an incredibly important step in bringing health into climate action. There's been a lot of discussion around decarbonizing health systems and making health systems resilient. So this is a very hot topic at the moment. I think, you know, there's an obvious part to this where you would say many low-income countries have contributed extremely little to the global carbon challenge that we have, and therefore why would they put decarbonisation as a priority? That is an important question. The same countries, of course, are really challenged by weaknesses in the health system anyhow, and poor finance, but also the impacts of climate through extreme events in particular. So there is an opportunity, I would say, through climate finance to actually strengthen some of the rural health facilities by ensuring that they have access to power from solar energy that not only makes them independent of the grid, or, you know, just dependent on microgrids, independent of diesel supply. It's actually a lot cheaper in Africa. Health facilities pay a phenomenal, well, all Africans pay a phenomenal amount for diesel, much higher than what you would pay in the West. And supplies are often disrupted, and it's an expensive need for health facilities, which obviously need electricity for safe and effective use of and development of their services. So the Solar for All initiatives to strengthen health facilities, provide clean renewable energy is kind of like a real win-win opportunity for the health sector. But I don't think it's the only one. I think that there's also just the whole idea of building resilient health systems that are not just about the facility themselves, but around the whole service infrastructure. And we've had some really interesting developments in terms of supply chains, for instance. Many of you will be aware that, again, most of my experiences in Africa, when the rainy season is there, access to health facilities goes down dramatically. The roads are may become impassable, bridges may get washed away. What would normally be a trip of, say, a couple of hours might take a couple of days. It becomes a lot more expensive, etc. So really access to health facilities, even if they're up and running, becomes really problematic. And there, innovations, for instance, using drones to deliver drugs and vaccines to rural communities, to rural and isolated health facilities, overcomes some of those really big challenges. So there are new ways that we can just improve health facilities and improve their resilience and reduce costs, etc. So I think it is a bit of an exciting time in that sense.
Yeah, and is it possible, Professor Noor, I'll bring you back onto this. Is it possible to achieve this while remaining equitable?
In the sense that if you think about the American health system, it's estimated that it's about 10% of the carbon budget of the US. It's a huge
the health system.
The health system. So decarbonizing health systems in high income countries is a significant contribution to global decarbonization. I think in low and middle income countries are choosing and trying to decide what sort of development pathway they want to move along. And as we heard at COP, there's finally an agreement to transition out of fossil fuels towards renewable energies, which in many places can be cheaper and more robust. I think there's really difficult what's the right way to say it? I mean, it's not a priority for low-income countries to focus on decarbonization when they have other issues to deal with. Really, the high-emitting countries, the ones with a high historical contribution, are obviously the ones that should be moving furthest and fastest. But the new technologies that are being developed could be very helpful for low-income countries anyway.
Cool. So thank you so much. This was a short question for us, but we thought it was very important. We see this all the time being mentioned. We know this is a mosquito masterclass, but malaria is a health system issue as well. So it was necessary to, kind of get the clarity on whether low-income health systems really have to prioritize this. And thank you so much for the clarification. So maybe all of us can go back to our wheelhouse, right? So the thermal biology of mosquitoes. So let's start with Sadia this is from a paper that I believe all of you co-authored. Fantastic paper by Erin on the thermal biology here, and you begin by listing these principles of the thermal biology of mosquitoes. We find this very important, and just for our audience, anybody who wants to get these notes, you can always reach out to us and to get them. But the question we have here is, could you guys just explain these species level differences that you see? The fact that a lot of the climate data has been collected, either on Anopheles gambiae or Anopheles stephensii. And yet, mosquitoes seem to be fairly different in terms of their peak performance or their peak optimum temperatures. So maybe you can hear from Sadia a little bit, and then Erin and Marilyn.
Sure. So one thing to sort of really think about is, as we think about where mosquitoes have evolved to be, they're sort of representing a deep evolutionary history to be adapted to where they are now. And so the parasites and the viruses and bacteria that they may be incubating that become human diseases that we then see in those locations. So we, ultimately we think things are where they are because we see a human disease case. And that's when we start looking at those mosquitoes. But in order to get, the data, Leah pointed out how absolutely excruciating it is to get all of these data. And we still don't know every vector, parasite, or, vector.
And then the parasites as well. Yeah.
Right. And so, for people who are doing the lab work side of this, rearing a colony of something that hasn't been reared as a colony, you have a burn-in time for that. And so we have sort of pieces of information from many different systems, but we don't necessarily have all the information from all the systems. But what this paper shows really, really nicely is that there is this whole range of different curves that are based on these sort of exquisite, pairings of the vector and its pathogen, and how that plays out across that entire life history. And, part of that is this question of vector competence, because it may be that you are, a mosquito flying around and you get a pathogen that you might then find growing in you. It's not that the mosquitoes are sort of, necessarily optimal when they're infected. And so it might be there is a fitness cost of some of the processes of being infected. And where that kicks in across the temperature curve is one of the more interesting things that I think we need to know more about too.
And do you think we should try and do it for as many species as possible?
Exactly, I do. I mean, I know that there's sort of evidence of mosquitoes being infected with pathogens because you can smash them and find pathogens in them. But actually doing the entire passaging process of demonstrating vector competence, where you have the mosquito not just infected, but able to infect an onward host, be it a human or not a human, that is a truly sort of, unique lab experimental setup. And to then do that for multiple temperature regimes is a very expensive and time-consuming process. And while you're doing that, of course, you're getting mosquitoes used to lab conditions. And so you've maybe already abstracted those mosquitoes from their field conditions. And so these are always going to be approximations of what might be out there, and sort of a first approach to thinking about what these parameters are. And I'm going to turn this back over to Leah and Aaron to talk more about sort of what determines the different shapings of these curves and how we derived the way we express them.
Yeah, go ahead. Thank you so much, Sadie.
I can take a first stab at that. There's actually some really interesting and nice theory developed by other folks besides us before we did this, where they examined traits not just of vectors, but across a whole range of primarily ectothermic creatures, and found that there are fairly consistent patterns in these shapes that we see of how different types of things that we can measure about organisms depend on temperature, and that some of them are unimodal and some of them are some not unimodal, some of them are symmetric and some of them are asymmetric, and they have some really nice theory about which ones are going to be which we saw definitely in the type of data that we have. So that's Priyanka Marisakare and Van Savage, and all of those folks that did some really nice theoretical work on those types of questions. And so we kind of used and built on that fundamental understanding in order to examine the data that we had. And so we found in general that we kind of see the same types of patterns, that the ones that we expect to be symmetric, things like survival, do seem to be symmetric in our data sets. The ones that we expect potentially not be symmetric, like in this one on the lower right-hand side, that's the parasite development rate and the mosquito development rate. So that's panel C on the left and panel D on the right, are tending to be asymmetric. There's some that there's not as good theory about which one it should be, and we can see that some of them, the data indicates that maybe it could be potentially either. In particular, because in some cases we have not very much data, in particular not across a broad variety of temperatures, and maybe not very many data points at each one, because at some temperatures it's really, really difficult to take data, which really impedes our ability to be able to understand what's going on. So we're able to kind of get an idea of these shapes, and we have different functional forms. In the thermal biology literature, there are literally like 100 different parameterized curves that folks use to describe how organisms react to temperature. And we chose some of the simpler ones because when you're dealing with cases where you don't have a lot of data, you need to keep it as simple as possible.
And that is before you add in the fact that the mosquitoes are also adapted.
Yes, I mean that's a whole other ball of wax. I mean, we can argue about
do you guys want to talk about that a little bit?
Can I actually jump in and go backwards one more, just make a couple more points on the previous slide? I don't want to cut off Leah though. So just in case you're not familiar with this and you haven't finished squinting to figure out what all this is, This goes back to that idea of a model of transmission being a function of many different traits of the mosquito and parasite. Each one of these little boxes is a different one of those traits. Each one of these lines is a different species of mosquito or species of mosquito plus pathogen for the pathogen-related traits. So if you kind of connect the lines between the panels, that's the same species, but different traits. So one thing that we found in this body of research is that there is this enormous consistency in those hump-shaped responses. And like Leah said, whether they're symmetric or asymmetric is also enormously consistent across species because it's fundamental thermal biology. And yet, if you look at these curves for different species, you can see quite a bit of variation across those different mosquitoes. So even though they all have hump-shaped responses to temperature, those are different hump-shaped responses. They have different peaks and different limits. If we go back to the previous slide that shows the overall R0, so the transmission model versus temperature, those kind of small differences in the traits across the life cycle add up into pretty large differences across different systems.
A lot of degrees of a difference,
Yeah, like if you look at the dark red, that's dengue and Zika transmitted by Aedes aegypti. It peaks at 29 degrees Celsius. If you look at malaria, that's kind of in green, that's in the middle, peaking at 25. And then if you look at, some of these more temperate viruses like St. Louis encephalitis, Eastern equine encephalitis, West Nile, they peak around 23 or 24. So we get a lot of rich differences in thermal biology that come from differences in these individual traits. And as Leah and Sadie were saying, it's excruciatingly hard to collect these data because it requires getting mosquitoes in colony in the lab, rearing them in a bunch of different incubators across different temperatures. And so for that reason, we often don't have information across the whole life cycle for a given mosquito species. And we certainly don't have population specific information. So you were just about to start talking about thermal adaptation. What this suggests, if we go to the next slide here, what this suggests is that a lot of mosquitoes are themselves going to be facing their upper thermal limits because of climate warming. And so the mosquitoes are going to be under selective pressure to adapt to warming. And that, to me, is one of the biggest gaps that we have in this thermal biology of mosquitoes work, is that we don't know how much adaptive capacity these mosquitoes have. And part of that is because we don't know how much existing populations vary in their thermal optima and thermal limits, just because it's hard to do those experiments and it requires getting all of those different populations in colony at the lab at the same time. And of course, there's so many other important research priorities that, it's hard for that one to compete for people's precious experimental effort.
No, I mean, big respects to the people who are doing this. This is why I was really appreciative of Nabi Bio's PhD thesis, and also the work that you guys have done. It's a tremendous amount of work trying to do this with the realization that two species might have different thermal limits, and if you're talking climate change, you want to be very sure that you're not missing a degree of temperature increase, because just 1 degree is going to be a big deal. At some point, we're going to bring in the conversation about humidity and rainfall and how that further complicates the whole thing here. But a little bit of a break from the difficulty here. You know what this organism is? So this is a sea turtle. And a colleague of mine asked me a question last week. Actually, two colleagues of mine. Joel Odero is one of them. He's a molecular biologist. He was wondering whether there is some kind of a genetic signature to these thermal tolerances. And my colleague Emmanuel Kaindoa works at Ifakara and was asking me whether it's possible to figure out a temperature at which mosquitoes selectively go a single sex. And there are organisms in literature such as this that prefer to be all male when the temperatures are very low, and they are all female when the temperatures are high. And I did find in literature that there are mosquitoes that behave the same. So we wanted to ask whether this is something that actually exists, whether it can be tracked, whether there is anything to it, like could you manipulate this thermal biology of mosquitoes for some kind of genetic manipulation?
So I will preface this by, this is really above my pay grade in terms of the biology, right? I'm not a mosquito biologist. And I know that in some data that we're working on analyzing right now, there does appear to be a non-even sex ratio in the adult mosquitoes that are emerging, and that this is potentially different amongst temperatures in the data that we're studying. So it's possible that it's there. I don't know whether or not this is something that we could manipulate per se, unless we wanted to do things like warm or cool bodies of water, right? I mean, because it's about the temperature during development. I think like, well, okay, I don't know. Maybe it's the temperature during development. Maybe it's the temperature of the adults. I think that there's a range of, I mean, so insects have really, really interesting biology that's very different between species. You have these species of parasitic wasps, for example, that usually like will be almost entirely female dominated, and you'll have like in a particular location a single male, that sort of thing. Because the mating systems of mosquitoes are different and they do the swarming behavior, I don't know how that impacts, like how the ratio impacts the ability of all of your females to become to be fertilized, right? So that they can produce eggs. So I'm saying that there's, like, I've seen some data where there may be evidence of skewed sex ratios that may depend on temperature, but I'm not sure how we could or would use that.
Thank you, guys. Thank you, guys. So maybe that's a point for Sandy. Did you want to add, or this we can mute
No, I'm
that
afraid
point for now?
that's beyond my knowledge base. But it's very interesting.
It's a very interesting one. You guys have already talked about this. Turns out mosquitoes can make a very specific adaptation to locations. Some incredible work here again, demonstrating that you can actually select for these adaptive features. So we won't belabor that. Let's go back to malaria a little bit. And now I will bring Madeline and Dave and Salam Noor back onto the scene. 2010, Nature paper by Pete Gething, Climate Change and Global Malaria Recession. Effectively saying that climate change will not lead to an increase in malaria in Africa, contrary to the claims. I need a commentary on this topic. Madeline, let's start with you. Is Madeline speaking?
Hi, can you hear me?
Yeah, we can hear you. What do you say about
Sorry,
this?
I just had to change my headphones.
No
So
problem.
I missed the question. Can you just give me the question again?
Yeah, so 2010, an interesting publication here, very clearly done, using existing data, climate data, and also disease data. End results, climate change is not going to lead to an increase in malaria in Africa, contrary to claims.
Yeah, so
your comment on this.
Can you hear me?
Yes, and actually they have their the blue figure at the bottom shows actually quite significant reductions in malaria despite climate change. Go ahead, Madeline.
So I think so there are two ways of looking at it. There's this one focus that climate change is basically directly impacting the dynamics of the mosquito, the parasite, and that you can model that, and by modeling that you'll get an understanding about how climate change is going to impact on malaria. That's one view. Well, I wrote a report with Hanan Nissan, who's a climate scientist for the WHO elimination scientific advisory group for malaria eradication, which was a discussion forum about what the impact of climate change was going to do. And one view is exactly this, that there are aspects of climate change which may actually reduce the transmission dynamics of malaria. But it kind of implies that climate change is only working on the biological system of malaria. And we know absolutely that malaria is really, the social determinants of malaria are huge. And so climate change is also going to be impacting and is already impacting on those social determinants. So when we think about, are we going to get more or less malaria as a result of climate change, we have to think about, are we going to have the same resources for malaria control in a world where climate is changing rapidly and many, many other demands are being made on finance? Are we going to have more or less malaria in a world where population movement is increasing dramatically and access to healthcare is reducing as a result? Are we going to have more or less malaria in a world where supply chains are being affected by extreme weather events? So I think this is to just have this very linear look and just run a model with temperature and rainfall and say that malaria is going to go up and down or down, to me, is not really going to address a real world situation. And yeah, so that's my initial discussion. I think it's much more complicated. If you have a look at the paper by Hannah Nissan on malaria eradication and climate, I think that provides helpful insights that climate basically impacts on all of the determinants of malaria. And so one should have a much more nuanced view about how it's going to drive. We know we had malaria in Europe before. We know we had malaria in the UK. We know we had malaria up to the Urals in Siberia. What has changed was socioeconomic development in many ways. And as socioeconomic development, where that degrades for all sorts of political, social, environmental reasons, malaria can return. So yeah, that's my opening piece on the discussion.
Thanks. Noor, in the beginning, you were talking about indirect effects. And I think that's what Madeleine has been emphasizing here. Indirect impacts. Do you agree with this view that linearly it might not increase malaria, but the circumstances still require a lot of attention?
I think, first of all, we've got to start from why we're asking this question.
Right.
Climate change is a massive problem for humanity. And a lot of the things Madeleine has mentioned, whether it is collapsing health systems or, some of the indirect effects are not malaria specific. These are things that will affect pneumonia. They will affect diarrhea. They will affect TB, HIV. They will affect all of health. And the response is that we have to mount is not going to be all because it's going to affect malaria. We therefore have to respond through a malaria lens. That's not what's going to happen. The socioeconomic determinants also work in a different way. Not just the disruptions that negatively affect society that will then negatively affect our malaria response. There would be urbanization, which overall will give us gains against malaria. There will be rural electrification. There will be greater connectivity. There will be broad socioeconomic development. All of these things will again have a reducing impact on malaria. And then we're missing the big piece, which is malaria interventions, which would have the most direct, largest impact on malaria itself. And if you look down the road,
I think that's the point they explained here.
Some of the anticipated interventions come through, then we are likely to see quite a bit of progress in malaria. But that should not be taken to mean that climate change is not a problem. So my concern really is, if we ask the question in a binary form, is climate change going to be a problem for malaria? You do miss the point. Because it's a problem for humanity. It's going to disrupt societies. It's going to lead to a lot of other problems.
So which question should we ask?
The questions you should ask is really, to me, look at what you need to do to strengthen the things that work with or without the climate change threat that we're facing. For example, the issues Madeleine raised around health systems. You don't need to have a climate change threat to know that you need to fix your supply chains. You don't need to have a climate change threat to make sure there's equity in delivery of care, and that you have sufficient health workers, and that poor people are not thrown into financial catastrophe in an attempt to get care. And these are just the fundamentals of public health, the fundamentals of health systems. And they're the very things that allow us to respond not just to the threat of climate change, but to threats overall. And so again, the question becomes, why is it that suddenly there's all of this interest in supply chains under the guise of climate change, when actually for nearly 40 years, 50 years, people have been saying we have a massive problem of supply chains, and that is impacting on access to quality care, right? So while we should concern ourselves, and we should, because it's a massive threat and it's going to affect humanity in so many different ways, and whether you are obsessed with the direct impact or the indirect impact or whatever it is that you're looking at, climate change is not going to be good for malaria because it's not going to be good for society. It's not going to be good for health.
Yeah.
So you got to step back and focus on what do we need to do. And what we need to do, fortunately, within the health space, are the things we've always needed to do to have a robust health system.
With or without climate change?
Without climate change.
Cool. Thank you. Thank you so much. Can
I come back in there?
Go ahead, Madhu.
So I think Noor has raised just fantastic what he said there. So I think what you have to say is like, okay, what is climate change going to make that is different from what we need to do anyway? And back in 2014, we had a meeting with the then executive team of Rollback Malaria in Tanzania, with the Met Services, the Malaria Control, and also the Ethiopian Met Services and Malaria Control. And what came out of that meeting to me was really interesting. This was 2014, so it's nearly 10 years ago, yeah? And the first thing was that what they realized as the executive team in Rollback Malaria is that the MDGs were going to end, the SDGs were going to come in, malaria financing was going to be more difficult going forward. And because of the siloed nature of malaria control under the MDGs, partnerships with the climate community and other communities had not been developed. And that this was actually put into the report, that if they wanted and expected to get the financing that was needed for malaria control, they would need to improve the financing, the partnerships then. And what we see now, and we see it at COP as well, is that many health programs, particularly malaria, just go, oh, malaria is climate sensitive, therefore we must be in line for adaptation funds. Yeah. And, you know, I think there are very good arguments to put malaria in line for
adaptation.
About that, for adaptation, but the partnerships are not there. So the trust is not there between the health community and the climate community. And I think that's a really important aspect, is that those relationships, it's not too late, but it's not a free lunch. It's not just like turning up and saying, oh, we need adaptation funds. It's actually really identifying what it is that is the piece that is going to support the whole adaptation community in a broader sense. And the adaptation financing is complicated to deliver. It's complicated to identify what is to be considered in adaptation, out, etc., etc. But the primary place is that the malaria community needs to see this as a partnership opportunity and not just as a financing one.
Thank you so much. I do hope that our colleagues on the line do appreciate these comments. A really important topic. We definitely can't discuss the entire piece here, but again, we will make this recording available and the slides as well if anybody wants to do that. So I'm going to push. Thank you so much, Madeline and Mr. Landwood. I'm going to push a question here to Dave and hopefully Leah. But before that, I just want to ask my colleagues from Glasgow or Ifakara if you guys have a burning question that needs to be raised. In the meantime, Dave, there's a question coming for you. Friends, is there a question we should pick up?
Yes, I have one question. Hello.
Yes, go ahead.
Sorry. Video and the microphone are coming from different screens, so I'm going to turn the video off. About climate change, obviously there are other issues that are related to climate change. One of them is food security, the way we produce food as mankind, and the way our crops are not doing as well. We're having less and less land to grow them. And in some areas of the world, we are trying to fight desertification by improving the way we farm into a more agroforestry way, so that we integrate a lot better our crop production systems with landforms that resemble a bit better the sort of local and native wild areas. So one of my questions is, if we are trying to fight desertification by integrating more wildness into our crop production systems, is that not going to bring more potential vectors into areas where humans are that otherwise would have been desertified?
Madeline, you want to take that? Thanks so
I'd love
much,
to
Eva.
take that. Yeah,
You're muted.
I know. Okay, so I think that's a really important question, actually, and something that's often ignored. I've done quite a bit of work, or in the past, on visual leishmaniasis transmitted by phlebotomide sandflies in South Sudan. Yeah, and these flies are really associated with balanites and acacia woodland. And once that became widely known, and it's a devastating disease that kills a lot of people in huge epidemics in South Sudan, some of the NGOs started chopping down the trees, yeah, because they act as potential breeding and are associated with the flies. And of course, that then is a disaster for the ecology. There's no, obviously we need trees. And I think what we have to say here is that we need nature. We need nature to be rich, diverse, and strong, but sometimes that can create risks as well. And therefore we need to figure out how to control those risks, as opposed to saying all nature is fabulous for health and all we need is more nature. Because of course the diseases are part of the natural system as well, all of the diseases that we get. So I think if we do have more diverse environments, some risks can emerge as a result of that. We just have to be aware of them and then build in the control opportunities to respond to the particular risks, rather than saying we're not going to build back biodiversity, stop desertification. Basically, a concrete jungle doesn't support a lot of life. It doesn't support mosquitoes, it doesn't support parasites, but it also
doesn't support humans.
Support humans, exactly.
Yeah. So again, partnerships there, Madeline. We need partnerships. Yeah. With the agriculture side. Is that Noor? Go ahead.
From earlier on, when we were so obviously there are a lot of lab studies that look at how different temperature ranges and temperature regimes affect different life-story traits of both mosquitoes and parasites and the system of both of them together. So one of the questions I always had is, if we want to bring this knowledge into policy that we then apply into the field, what is the ideal timescale at which we want to look at temperature? Obviously, an average temperature for a country throughout a year is not enough. But knowing the temperature in 1 centimeter square every second is a bit too much. So what's an ideal time and spatial scale that we want to care about in order to then bring what we're learning from the lab into policy for the field? Because right, that is one of the most interesting goals of doing this.
I believe I heard someone say spatiotemporal scale, and therefore my ears perked up.
Go for it.
So I think it's a really key question here, is thinking about sort of what the timescales are within the mosquito life cycle, what the timescales are for parasite development, and what the experienced temperature regime is of those mosquitoes in their natural habitat. And then the question really is, can you replicate all of that in a lab experiment? And the short answer is no, you can't replicate it all. But I think when we're talking about measuring things in the field to approximate what we can know in the lab, in the lab, when we're doing these constant temperature experiments, we're sort of assuming, an ideal world, right? And so if you're keeping things constant, a daily temperature in the field is a constant day, but you won't get the whole mosquito and parasite life cycle within that single day. And so you can probably create some approximations if what you're trying to do is re-replicate the lab. The interesting parts are where temperature variation occurs and what that means. So, a lot of the sort of both the math and the lab experimentation now is asking this question about if you get outside of your sort of predicted optimal range, as it were, or even your thermal boundaries, depending how long you spend outside of it and for how long, and how long you as a mosquito population experience that. And that's what's in Nina's new paper. How much tolerance is there at those sort of shifts, and can the mosquitoes themselves adapt to that shift? And so to go back to that question about sort of the spatiotemporal scale, for Aedes aegypti, I know, we tend to think, oh, there's this 250-meter range because somebody published it somewhere once. We've sort of, looked at different questions about whether 100 meters is really how far Aedes goes in a lifetime, or if it gets picked up by the wind, it's going to, go a long way away. Does it then complete that whole one mosquito life cycle all that distance away? And that's where you start getting into these sort of, what's the spatial scale at which you want to interpolate temperature, if you're just talking about temperature. And so I think this is a really important thing to be thinking about, especially sorry, somebody in the chat also brought this up, I think, asking this question about, are we talking about air temperature or surface temperature? Are we using Earth observation data to be thinking about what we're measuring versus should we put microclimate loggers every 50 meters everywhere in the world? I think, all the computers would explode if we did that. But I think, somewhere between that is a really sweet spot of really big data that we can start to harness. And, there's lots of data fusion methods now, so we can think about being able to actually use our longer term time series in conjunction with satellites that are only now beginning to, have their missions complete enough for us to actually have those time series from satellite imagery. And so I think there's sort of this key moment now in the spatiotemporal climate data availability that we can start to then think better about matching that to the biology of these mechanistic systems that we're currently relying on lab-based data to deal with. And I will kick this back to the people who are closer with the lab stuff.
Yeah, no, definitely. Also, now that you bring the questions that were in the chat earlier, someone was asking as well about the impact of daily fluctuations. And I mean, all life on Earth has evolved to deal very well with circadian cycles. And the length of day, it changes slightly with time, but like it's pretty much constant throughout an individual's lifespan. So it makes sense that not all biochemical processes happen at the same optimal temperature. Actually, organisms that regulate their temperature have different body temperature for different processes. We mammals sleep and we have a different body temperature while we sleep and while we digest food than while we run away or fight or whatever. So it makes sense that both the mosquito and the parasite are adapted to an oscillating temperature throughout the day. So also, if during the day, for example, we visit certain extremes of hot and cold temperature, then there's also a behavioral adaptation to that. You can either run away from extremes or just withstand them because they won't last for that long. So when we do all these experiments with temperature, is it okay to use constant temperature? Sorry, someone saying in the chat, I am muted. Yes, I'm in the same room as someone else, which is why you can hear me through their microphone.
We actually do have I could skip that directly to that if someone wants to answer that question, because we do have the fluctuation question raised. Is it better to keep these things at a constant temperature, or should we go for the fluctuating?
I'd like to comment on that, if that's okay. I think there's been a real trend towards measuring in laboratory experiments, doing these what we call DTR experiments, daily temperature range, where people fluctuate temperatures instead of just holding the incubator at a constant temperature, because the perception is that's more realistic. I mean, it is more realistic for what mosquitoes face in the real world. The problem with fluctuating temperature is that if you're particularly interested in just that one temperature regime, like you're trying to model your lab experiment exactly off of the temperature regime in a particular place in the world, then that type of design might make sense. But if you're trying to use that to understand the broader thermal biology of a species, you need to know something about performance across a range of temperatures. So, we showed all those nonlinear thermal performance curves earlier. When you fluctuate across varying temperatures, you could think of it as kind of integrating over that hump-shaped curve. And so sometimes when you integrate, you're going to be integrating over a part of the curve that's not linear. So performance might be lower or higher than you would expect if you were just holding the mosquito at that constant temperature. But in order to know that, we need to have a constant temperature thermal performance curve first. So I think a mistake that people sometimes make is they will measure mosquito performance at varying temperatures and at a matched constant temperature, but we don't know where we are along that thermal performance curve. And so it doesn't really give us enough information to interpret why did the mosquito respond to varying temperatures the way it did? Because we don't know, well, are we on the linear part of the curve where temperature variation really isn't going to have a big substantial difference, or are we on a different part of the curve that's more or less nonlinear? So I think both sets of experiments are important, but it's important to kind of couch those temperature variation experiments in thermal biology that let us understand the full extent of a mosquito's temperature response across a wide range of temperatures and across different parts of the life cycle. I know that's a tall order because we keep talking about how hard these experiments are, but I think capturing that thermal variation is important, though.
And I'll add to this, this is actually a general open question in thermal biology, not just for mosquitoes. Like even for simpler organisms like fungi or bacteria, we actually don't do a very good job of predicting what's going to happen when temperature varies based off of what happens at constant temperature, let alone like generalizing from one varying temperature to another. So there's actually a really interesting open gap for all of you graduate students, especially that are interested in these sorts of things, like how, what is it that we're missing in our understanding of fundamental thermal biology across multiple types of organisms that is making it so that simple integration isn't always doing a great job of predicting. I mean, it's not just mosquitoes, like there's some really interesting work that folks are doing on like lizards. So there's still like these really interesting open questions about how we should be actually like building our models for varying temperatures.
Can I add something in here?
Yeah, very quickly. Go ahead.
So I just think that well, first of all, the question I think was around the scale that connects the biology to policy, and I think that bit has not. We haven't got that discussion going. But I think also about even on the modeling side. When I was at Columbia working at the International Research Institute for Climate and Society, I really became aware of how the climate community moved from developing single models, some mechanistic, some statistical, and eventually created these big multi-models because they knew that each model actually was not very predictive. None of them were that good. But once you started bringing them together, the bad bits of each model started to cancel each other out. And the stronger bits of the model started to become more, what's the right way to describe it, would add value to the other models. And that's how modern probabilistic weather and climate forecasting is done. I just think that on the mosquito side, we're not there yet. And in part because we don't really have a strong connection yet between the biology and the operational use of these models. And the operational use of these models really requires an understanding of the appropriate scale of decision making. What is the time frame? What types of decisions are being made? What is the appropriate scale of decision making? And I just want a question I have for everybody. This year we're in an El Nino. It's the biggest signal we get on temperature in the natural cycle of things. Around the central belt, we'll have extremely high temperatures this year and next year, and then hopefully again it will dip down again and maybe we'll go into a La Nina period. If our models are any use whatsoever, we should be able to exploit these natural experiments and show which parts of the world do we see an uptick in malaria that seems to be associated with temperature, which is definitely going to be in the highland areas or in the cooler margins of transmission, and in which areas do we not see that, partly because in many parts of the world, that warming is going to be associated with drought. And so you'll get, like people said earlier on, this conflicting issue between, it's not just temperature, it's actually what's happening with rainfall and humidity at the same time. So I just thought I'd throw that in there as a.
Do you have an opinion about that, Madeline?
Opinion as to
what are your own thoughts on that?
Those, so
we just picked up your quote from this about possibility of seeing the thing we don't want to see.
Yeah, so I think I think there's a lot of value in forecasting. I think that we need to develop forecasts that are really initiated around the questions, and there are lots of questions around. It's not just let's predict an epidemic, but let's just see the evolving risks that are changing in the climate on the one hand, with the impacts on malaria on the other hand, the likely impacts, and they're quite crude. I think you're not going to get decision makers using very specific information, because that's going to be so location specific. They're going to be looking like, what about the north? What about what's going to happen in the highlands of Ethiopia? What's going to happen across the Sahel band between so many millimeters and etc. What's going to happen in coastal Tanzania? Those are the types of environmental structures that we need to be able to predict against.
And on that note, I think I'm going to cut short the discussion here and bring it back, because that's the topic we're heading to. I want to bring it back to David. I see our time is almost running out. I want to bring it back to David to help us move into this highland malaria question. So Madeline, stay on standby, and we're coming back to you. But first of all, Dave, many authors say that the greatest impact of climate change is going to be on the fringes. We read this in literature repeatedly. And yet we also see this situation where we have a growing problem in the highland areas, the places that previously had non-stable malaria or non-stable transmission, certain transmission. The origin of this story is from here. At least the first paper we read it from. This is a Frank paper from South African malaria. I don't remember the full name of the organization, but the South African Malaria Research Organization. And when they did this model, he was very categorical that the greater burden is going to be upwards rather than lateral. There's going to be an increase in malaria, but it's going to be altitudinal as opposed to latitudinal. So that brings us back to this question, and I just want to ask Dave to tell us what your understanding is in terms of where climate impacts are going to be greatest. Should we worry about these internal perturbations that we may not be paying attention to, just because they're deep within the tropics, or should we stay on the fringes and look at how the latitudinal changes are happening? Back to you, Dave.
Wow, I feel a little bit put on the spot by this question. So let me.
Anybody can take it, but you start.
Let me give some historical background. So that paper that you were just seeing, there was a big movement in the sort of 25 years ago to try and build these climate models that were predictive of malaria. And then what happened next was that there was a big movement, and this was led by Bob Snow and Simon Hay and Carlos Guerra, to collect as much data as they could. So that led to the creation of the Malaria Atlas Project databases, and Noor was, very centrally involved in all of this. I was peripherally involved, and the collecting of the malaria data ended up sort of leading to these the use of Bayesian geostatistical models and the creation of these very high quality environmental layers to try and actually put all of this to the test, to say, can we predict the distribution of malaria based on environmental covariates that are uniformly available. So the game that Malaria Atlas Project was playing was that we can only use an environmental covariate layer if it was available everywhere. And then there were these sort of extensive efforts to try and explain distribution patterns based on those factors. So one of the things that has come out of that that sort of was very interesting is a really, and I'm going to come right to the heart of your story is this, is malaria and climate change locally related, on the fringe areas, is it related to changing climate or is it something else? So there was a big debate, and I don't want to get into all of the details, but a guy named Dennis Shanks went and collected the data from some tea plantations in Caricho, and they started doing some analysis. And initially, they were convinced they were going to find that temperature was responsible for it. But Dennis is an MD, and Dennis was really convinced that it was really during the 90s, during the period when this study was conducted, that it was a changing frequency of chloroquine resistance that was really responsible for the big patterns. And so there was a debate. So Simon Hay and others published this study and an analysis of the data, basically pointing out that, there wasn't much of a change in temperature over the period. And there was a debate about the the stationarity of the temperature data set that led to some nasty name calling. But essentially what happened thereafter was Dennis went back and got a few extra years of data. So when chloroquine resistance, when the drug policy changed, temperature kept going up, but malaria didn't. And so, what is it really temperature that's responsible for changing temperatures at the margin, or is it something else? Is a really, really hard question. And I'm going to say this, I'm very skeptical that we have any hard, really, really well documented stories that temperature changes have actually driven malaria up a mountain. And
we do have sorry to interrupt you there, but we do have, we put up two papers here. The first one is the paper from Usambara. I think this is the most commonly quoted from Tanzania, which illustrates significant decreases in malaria as you go upwards. And then we have this paper from Madeleine, actually. Thank you so much, Madeleine. We got so much of your work. We are grateful for all that you've done in this field. But where she actually demonstrates with the team there that you do have a temperature change. So this is plotted as a elevation vis-a-vis the temperature changes over the years over the elevation. So kind of, ties in the question of temperature to the altitude, so that it's not just altitude on its own, but temperature. And then we have, of course, the Usambara data, which, as you can see, has most of the malaria indicators declining as you go up as well. So back to you, Dave. And it would be really nice to hear what your explanations for these are. If it's temperature, or if temperature is just one of the covariates, or if something else, that would be really nice to hear from. Back to you.
So I'm not gonna touch Madeline's paper. I'll let her discuss it. But if we can go to the Bodker paper, I just want to point out that, so I sat at a bar and talked about this paper with Steve Lindsay and his frustration that when people cite this paper, they ignore the fact that there's a conclusion in there that the dominant, yes, there was a gradient in temperature and all of that, and there was a gradient in malaria transmission as you go up and down the mountains, but that it was probably hydrology. It was the amount of available water that was really explaining differences as you go up and down the thing about how much malaria there was across this thing. And I think it was interesting to me to hear Steve's frustration about that, because he thought that there was a much more subtle story here, but it sort of told the story that people wanted to tell, so they cite it the one way rather than the other.
So when they cite it, they talk about temperature,
only temperature, and they're missing the
because
nuance.
in this same paper they say that at least in the upper areas the transmission there is non-stable. So it could be the hydrology thing you're talking about. Back to you, Dave. Sorry.
So there is a real effect of temperature and altitude on malaria transmission, and it's one of those many factors affecting malaria transmission. But if you sort of went to any particular place, like in Caricho and the tea plantations, you have to also pay attention to what people are doing. It's not just the mosquitoes. So in the tea plantations, this is a highly skilled labor population that's there for work. And because it's a skilled population, they put investments into people, into the health system. So these people were going home for the holidays and coming back with malaria and with chloroquine-resistant malaria. So that's part of the epidemiology story in the Carichos that you have to sort of weigh against all of the other analysis that was done. In this case, I wish Steve could speak for himself, but yeah, I'm going to shut up there and let Madeline address her own paper, because I think.
Yeah, well, what I think is you're pointing out is it's just impossible to make proper scientific inference from trends. You can't do it. Yeah, it could be more malaria because the economy's collapsed and people no longer can access anti-malarials. It could be less malaria because girls are better educated and know how to. So there's trends are hopeless in terms of trying to look at causality, which is why I focus so much of my effort on sea surface temperatures and El Nino, because it's one thing where you get these really big signals in the climate system that are short term. So when you have an El Nino, you get warming around the tropics for maybe a year, a bit longer than a year, and then it will start to go down again. When you get a La Nina, you tend to get a cooling period around the tropics. So you've got some quite defined windows where if temperature is driving stuff, then it should be more likely to increase when you have an El Nino and more likely to decrease when you have a La Nina. We have the confounding aspect of rainfall. So when you have an El Nino, some places that usually are normally wet become dry, and places that are normally dry become wet. When you have a La Nina, you have places which are normally dry become drier, and places that are normally wet become wetter. So there's some structure to the way the climate works. And my thing is that we have to exploit the natural experiment because that's really virtually all we've got. So just looking at trend lines is just not going to get anywhere. And so the paper that we wrote in Botswana back in 2005 is very clear signals with malaria data once it's detrended for risks around chloroquine, etc. Very clear signals that ties the case numbers to sea surface temperatures. The same has been done in Ethiopia by other colleagues with Mercedes Pascal and Siraj. So that gives you a very clear, when you have warming in the highlands, in parts of the highlands, but not everywhere, you get these bumps in malaria. Now, why don't you get it everywhere? So I just wrote a paper with Adonia Wayesa from the Ethiopian Public Health Institute. We tried to put all of the very messy data together in an organized way, and it's very difficult to see what the conclusions are.
Do you want to talk about that? I think we have that here.
Oh, yeah. So I just also going back to what David said about the origins of the Malaria Atlas Project, etc., and the types of climate data that are available. They're incredibly crude. So you would go to a village and identify malaria from a very specific locality and then try and associate it with a very crude climate data set, which has very little observational data in it. So understandably, you're not going to get great relationships. And one of the things that I found is that over time, the better the quality data, almost invariably, the better the relationship. I.e., if you see a vague climate signal in a malaria data set and you improve the quality of the climate data, you get a stronger signal. And I also you see that in the way climate people do predictions and what have you. So one of the things I worked over the last, 10, 15 years is to try and improve the quality of the data available to national decision makers based on not only all of their own observational data, but integrating that data with global products. So they have a better quality data set to start with. And that was the main piece in the most recent paper, is that it's possible to produce much better data that can be used by national decision makers. That is going to be better than FuseNet data. It's going to be better than ERA5 data. It's going to be better than the types of products that we use in the Global Atlas. It's still just a beginning. We're just scratching the surface of the data issues from the climate side that we need to sort out in order to make it really useful in terms of getting climate into health decision making.
Well, and if I could kind of add on to that, in addition to kind of the data issues that Madeline brought up, there's the data meets models issues that we have to really think about as well, right? Where if your predictors are uncertain, like they are for the climate, there's a really nice statistical fact that when you do a correlational model, you're going to, if you have uncertainty in your X's, the things you're using to predict, you're going to bias your estimates of any relationships that you have. And typically you're going to bias them towards zero. So that is, you're going to tend to find that you'll be more likely to say that there's not a relationship when there is. Further, like there's a whole kind of underlying thing, again, with our data where our predictors are correlated. And again, when our predictors are correlated, we're going to be less likely to be able to get estimates of any of our relationships between our predictors and our responses that we're interested in. And in this situation, we also have an additional confounding factor that where the environment is good for transmission, that is where we are intervening, right? So you are artificially decreasing in cases in places where the environment is good, and you're more likely to be doing it there than places that are marginal, right? So you actually can change your signal through our interventions. So we should expect that it's going to be really difficult to kind of identify all of the identify well the factors that are influencing the climatic factors, as well as other things, right? I mean, if you're intervening in places where there's a lot of transmission, those places might be areas where the socioeconomic status is lower. Like there's lots of these things. So it's always going to be much more difficult to actually just purely from a data and modeling perspective to be able to actually infer what those relationships are like. Now, in some cases, folks would, for other systems, folks say then like, oh, then maybe we shouldn't intervene. That is not an option we have for something like malaria, right? So we can't change and adjust our experimental design to enable us to tease out these factors. We've got what we've got, right? And we frankly probably need more intervention in many of these areas. But this does lead us to be able to think about, like there was stuff that you were asking about in the margins. We might expect that in the margins, we might actually just be able to tease out factors slightly better because we have less intervention. For example, although there's other difficult aspects of that, because effectively, as long as you get enough, like your climate is good enough, you're going to be able to have enough because of some nonlinearities between kind of transmission and prevalence. As long as you get kind of enough transmission, your prevalence kind of saturates, which means that you don't really notice like small differences where things are good. But you might see that at the edges. But if you're at the edge and you happen to intervene, like you're gonna just not see anything. So these things are not simple. And we
one thing that we would really, I think one thing that would really be very helpful, maybe it's a difficult assignment, but just based on what you guys are saying, one thing that be a really fantastic outcome of these conversations and maybe follow-on conversations is if you guys could help us, figure out what would be, hypothetically, what would be the perfect design for for these data collection systems that would capture the disease pattern, the climate pattern, the mosquito patterns, the genomics patterns, if any. How would you design such a system? So, I mean, maybe at a later date, this is a conversation we could have, but it looks like the Holy Grail.
FREDROS, can I come in just to follow up on the conversation I've been happening?
Yeah.
We've talked about the way the strongest signals of the effect of climate change come from the margins of transmission.
Yeah.
And if you look at all the countries that have highlands where there is some form of malaria, these are the areas they also see as offering them the best chance to eliminate malaria. And so these are the places where they have identified as the places where they're going to get rid of malaria. And in some countries, there is intense actions to get to zero. And so the question then is, from a programmatic perspective, if you go to a program today and you tell them, look, climate change, the strongest signals, the biggest impacts are likely to be on the margins. And then they tell you, actually, it's in the margins that we are making the most progress. And it's the margin in the margins that we've identified for
But
elimination.
you mean yeah, epidemiological margins, not geographic margins.
No, it's the epidemiological margins, like where the transmission is lowest. That's where
lowest. Okay.
For elimination. And so you end up in these situations where it's very difficult to communicate to programs what is actionable, right? From the perspective of responding to the climate change threat. And then a lot of issues were discussed around the data. And remember, you don't want to go into a country to start advocating for strengthened data platforms because of your concerns around the threat of climate change when you're actually having difficulties articulating that. That conversation should happen because strong data systems are essential to a massive enterprise like the malaria enterprise. And that particular
public health enterprise in general. Yeah.
That particular discussion hasn't been successful for a number of reasons. And it's unlikely to be successful because suddenly you've come in and you said, climate change is a massive threat. We need to collect the right information to answer these questions. The systems don't respond like that. There has to be another imperative. And that imperative has to be, actually, we need to collectively respond to all the major public health priorities. And therefore, we need the right information platforms to do that. If that then allows you to get the right information to address some of the more kind of experimental questions, that is fine. So, in communicating this, we've been faced with a number of challenges. Where we're saying climate change is the biggest threat, is where we're seeing the most progress, is where countries are aiming for elimination. And we just have to bear that in mind.
Yeah.
Yeah.
But we are no longer shrinking them up.
Can I add something?
Go ahead, Madeline. Yeah.
So, yeah, I know. I think that's really helpful. And I think the way, to me, is to position climate and malaria as in a really structured way. So, yes, we need all the different types of data. But first of all, if countries are not taking into account the seasonality of the disease properly, they're going to miss opportunities for control. And a very good example for that is Ethiopia, which has a very complex seasonality. Yeah, there are 3 different seasons. They vary to a certain extent in different areas. You see when NGOs come in, sometimes they just treat the whole country as one country, or they say there's 2 seasons, but no, there isn't. You have to know what's happening locally, and you have to know the seasonal structure, because interventions are often impacted by when you undertake them relative to the season. So that's one thing. The geographic extent of the disease, where are the margins? You can map that. Understanding where you get epidemics and the development of early warning systems, that fits in there. That's all basic malaria control, which should be routine in every country. Then on top of that, you get trends that you want to understand. And as I was arguing, you need to be able to understand them with variability. You can't understand the trends by themselves. And then the other one that I think is really important, and it was the one that was the one time that I felt we could get really senior policymakers paying attention, is when in Tanzania, the UK's PMI programme obviously has put in very large amounts of money into malaria control. And this whole question in East Africa, they were raising, they're going to report back to Congress about the declines in malaria in Eastern Africa. And in Tanzania at the time, they set the baseline year was the 5 years prior to 1999. Yeah. So, which included a huge El Nino and major epidemics all over the country. And that was going to be the baseline period. And then the control period was a period where there had been 3 mega droughts. So my concern was, for a policymaker perspective, if somebody says you've spent £5 billion on malaria control and you don't know whether or not those droughts are accounting for 40% of your decline in malaria or whatever it is, then you are putting your budgets at risk. So actually really understanding what baselines are, understanding how climate impacts on the outcomes is very important in terms of taking it as a counterfactual and being able to articulate, drought might have done X amount to malaria, but we know another X percent is due to control, because of course those droughts will presumably end and your rains will come back, and then people will think, oh, it's a failure on the control program, but it may be absolutely no failure at all. Yeah. So just having a really structured approach to the types of decisions, the timing of the decisions, the information that you can use from climate, as long as malaria decision makers actually can incorporate it into their planning process. And that climate change is one of 5 things that you can do, but they all needs to be integrated into one kind of package.
Can I jump in quickly with a sort of thought on this question about sort of how we're talking about climate models and then how that translates up to sort of the decisions in the mapping stage. I think there's sort of some key terms here where climate can limit things versus climate driving things. And I think when we start to have this conversation about sort of things in the middle and at the fringes, we really need to be thinking about, there are these multiple layers of factors happening, and climate, this thing we're calling climate without defining whether we're talking about temperature, humidity, extremes, averages, or all of the above, right? It's, we've got a latent variable called climate. But I think sort of where we can detect trends is where we're seeing sort of that driving, but then where we sort of find these boundaries on the edges of ranges is really where climate can be limiting the processes we're talking about. And the decision making we can make, right, can be based on the events and sort of, rapid response, or they can be based at sort of the beginning end. Are we forecasting that things are going to happen? What would you actually start to implement at that point? And so the scale at which we want to do these sort of forecasting and planning experiments depends on the scale at which even those decisions are being made and what they can be. And I think that sort of speaks a bit to what Madeline was saying, and the slide that had, how do we deal with early warning systems? When do you alert someone? What do you do as a response to the alert? And for a sort of juxtaposition of, the situations we're all probably picturing in our heads versus perfectly suitable malaria area, this summer in Florida, there were a few cases of malaria and everybody said, is this climate change? And if you look at Florida, there are year-round competent vectors for malaria breeding, but there's also immense response and control, and has been for a long time. And I think that sort of gives us this, this lens on climate is a layer over the top of what's happening. And in places where it is suitable, didn't stop being suitable, it was just really, really strong intervention.
Right.
How do we use these as our sort of case controls for thinking about this issue of place and the sort of frameworks in which we can do intervention versus space, when we're saying these are conditions that could exist in all these places. And I think there's sort of, we need to be more clear about that, but I don't think it's really a tension. I think sort of that would be a false dichotomy in these cases.
No, thank you so much for no problem. Thank you so much, especially for the last bit there. We all need to figure out a way that the integration happens, that the partnership happens, that the people are cross-fertilizing these ideas. I mean, when Madeline was saying that most counterfactuals at the moment do not necessarily include this adverse, occasional drought, then you start to wonder, because those occasional droughts or occasional El Niños can actually have such quite a big impact on disease front. So I want to hold this conversation there, and because we have just a few minutes left, but I really do need you to stay with us if you don't mind. We're going to talk about just 2 other things, and then we will close. I might reach out to you guys to request a phase 2 of this masterclass, because it's really, really interesting. And we were going to talk about these extreme weather events. We just covered the highland malaria situation. Extreme weather events, of course, when my colleagues were giving the overview in the beginning, they did tell us that the extreme events are becoming more frequent. They're coming to new locations. So there's there's all this understanding already as to why we need to pay attention to them, and people like Madeline. I actually wanted to ask you, Madeline, if you realize this goal C1.5.
Sorry.
At least temporarily in some place. El Nino is still going on until 2024 mid. 2024.
Yes. Yeah.
But it's already quite a massive situation. And if you don't mind, if you could just do two things for us. Number one, provide a very basic explanation for El Nino. And number two, explain the differences in impacts and why we should worry about it vis-a-vis vector control. And all our other experts can jump in, please, if you don't. You're muted, Madeline. You're on mute. Yeah. Still on mute.
Okay, can you hear me now?
Good. Yeah, now we can hear you.
Yeah, so it's always good to have a climate scientist in the room when we talk about climate. And this is one of the things I've learned. I'll do my best, but
It's okay.
what I would say.
You're muted again.
Can you hear me?
Now it's good, yeah.
Okay. The El Nino Southern Oscillation is part of the natural climate system, yeah? And it's basically an oscillation in sea surface temperatures and first of all, everybody is, I'm sure, aware that the climate system basically, or if we start off with the weather system, meteorology focuses on what happens in the atmosphere, yeah? So it's very localized. It's what happens in the atmosphere. Climate, when we want to look at seasonal climate or climate over 5 to 10 years, shifts in climate or long-term climate change, that's really an integration of what happens in the atmosphere, what happens in the sea, and what happens on land. So it's really the global climate system that works together. So with the El Nino Southern Oscillation is this natural fluctuation in the climate system, and it's got three phases, if you like. El Nino, when you get warming in the Pacific, and you can see on which graph, maybe on the graph on the right where it says wet and warm. So in the middle of there is an area of sea where when the temperatures rise and they are maybe half a degree above normal, they start to influence the atmosphere above them. And that influence starts to shift the direction of the winds, because at that point you've got winds, which are the trade winds, which I can't remember which they go from right to left or left to right. But anyhow, it starts influencing the direction of the trade winds and can actually switch the direction of the trade winds. When you have a La Nina, which is the cool phase of El Nino, that area off the coast of Peru tends to get cooler. Yeah. And what it does is it intensifies the winds. So it brings more rain to the wet areas, more drought to the drought areas. El Nino tends to flip the climate, and it's really dominates around the tropics. So you get very strong effects around the tropics, much weaker effects around in the extratropics. So, yep.
Yeah, go ahead. Yeah.
So it doesn't affect everywhere equally. I can't say that this is the best graph that I've seen, but what you will get is strong effects in not just in regions, but in seasons. So for instance, in East Africa, there are two seasons. You have the March, April, May rainy season, which is the main long rainy season, and you have the short rains, October, November, December. El Nino will change the short rains, and we've just had very heavy rains in Nairobi. Somebody can tell me if that's not the case, which are considered to be associated with this El Nino. Yeah. We had a long period of drought in Somalia, which will have been associated before the El Nino. There was a long La Nina period, cooling period, and that resulted in excess drought in East Africa again, particularly in that short rains. So you have to think about El Nino is change in the climate or an intensification of the climate when you have a La Nina. And when you have certain regions very affected, but not all regions, certain seasons very affected. So if you only have one rainy season, like in Southern Africa, that is very affected by El Nino. We tend to get droughts when you have an El Nino in Southern Africa, and it tends to be wet in East Africa in the short rains. A country like Malawi is half in the East Africa picture and half in the Southern Africa picture. So in one country, you might get the north is wetter than normal and the south is drier than normal. What's good about El Nino, if you like, what's positive about it, although it tends to be a bit more of an extreme climate, is that from the 1980s, people understood you can actually use it in prediction systems. Because sea surface temperatures stay warm for a long time, once they've started warming up, you know they're going to stay warm. And therefore you can say, aha, in a few months' time, when the new rainy season start or the short rains start for East Africa, we will expect them to be wetter or drier. And the same thing for Southern Africa, to a certain extent for West Africa, very strong signals in Asia, very strong signals in the semi-arid regions of Brazil, for instance. Yeah. And because it gets really strong rainfall signals with the wind, but also across the whole region, a bump in warming when you have an El Nino. Is that helpful?
That's very helpful. Maybe just to complete, just before you go, you work at the Wellcome Trust. The Wellcome Trust is very, very active in this field and has begun this incredible program to help scientists and countries really generate the necessary data to understand climate. And on behalf of my colleagues and everyone else, we have to say a big thank you to the Wellcome Trust. I wonder if you wanted to use this opportunity, maybe 1 minute, to say a little bit about what you at the Wellcome Trust and your colleagues are most interested in going forward in this, and why this is an important point of focus for you guys. You don't have to, but just in case you want to.
Yeah, very quickly, we're focused on, I lead the climate impacts and adaptation work. And if we're going to adapt to a changing climate, we have to understand the climate we're adapting to. That to me is a kind of no-brainer. Otherwise, it's business as usual. So it's very important then for the health community to become more engaged with understanding how the climate works and how it drives disease and how it will shift things one way or another, and how the long-term trends interact with the natural variability, because that's actually how we'll experience the climate in the real world. And that's what the control programs will need to respond to. So far, we've invested particularly in heat research, and we've invested in heat physiology and in heat adaptation. Our colleagues who are focused on infectious disease have already invested in climate data and disease modeling for decision making through the CSID call. I can see there are people here who are benefiting from that and participating in that process. We're definitely going to be building up, and a key interest for us at the Trust, and historically, and still, we are interested in a mechanistic understanding the causality, understanding the drivers, but then also how do we make that practical and real to decision makers? And that's quite a long stretch between those two things. And what we're trying to do is create investments that help bridge that gap. But given the urgency of climate change and the challenges that many are facing already, we're trying to focus on a few key things. Our infectious disease colleagues are bringing climate into the work that they do, and we're supporting them. Our mental health colleagues are bringing climate into what they do, and we're supporting them. And we in the team at the moment, we have a strong interest in the impact of heat per se on a number of things. We're doing physiology, but we'll also be doing mental health. And of course, we've had descriptions here about the thermal performance curves of all living organisms fundamentally are going to be impacted by heat and changes in heat around the world. So so we are here. We always look to great ideas, which is one of the reasons I'm very happy to join this session and listen to everybody and learn. We want to understand how we can be most effective in protecting vulnerable populations around the world from changes that are happening faster than we anticipated.
Thank you so much, Madeleine. Thank you so much. Truly appreciated. In the next, we have only 15 minutes to go, and we're going to cover two things. Number one, we would like to talk a little bit about dengue fever and Aedes. And number two, we would like to talk about humidity and hydrology. My voice is dying here, so I'm going to request my Glasgow colleagues or Ifakara colleagues, if anyone wants to help me out with the questioning. But Dave and Sadie and Erin and Sadie, first of all, before that, just to close this chapter, La Nina, El Nina, extreme weather events are not just biological questions. They're not just weather questions. They also have big impact on the economies, and especially for these low-income countries. And we have this example here from Peru. If you look at this data, it's to the tune of trillions of dollars. So you can imagine that if you had a good adaptation process, the return on investment is probably quite big. But if unmitigated, some of these programs, be it like the cyclone in Mozambique or the El Nino events, they can be very costly for low-income countries. So for the scientists working on this, I think this is an important subject. You need to keep going at it, and just to realize that beyond the health challenges, there is quite a big economic problem here as well, which trickles down or has negative externalities on public health as well, and we need to be aware of that. And Noor was already explaining this earlier. Some of these impacts, we're already seeing them directly. We could see in the World Malaria Report the situation from Pakistan, where all of a sudden nearly 2 million malaria cases coming in. So again, very real problems here going forward. I think Madeline mentioned already sometime when she was speaking earlier, the work that she did in response to the SAGME report, WHO malaria eradication report. How do you climate proof your malaria strategies? So again, you're welcome to read all this going forward. So on that note, I would like us to, first of all, before I shift, Dave, Dave, are you still there? I hope Dave is still with us.
I'm still here.
Yes, we hope that you can stay a little bit longer because we have some questions for you regarding the humidity question, if you don't mind. But before that, I would like to request Leah, Erin, and Sadie, if you guys don't mind, we want to discuss a little bit about dengue fever. You guys have written that, climate change might not necessarily increase malaria. Forgive me for using that phrase, but it will probably bring dengue fever to Africa in a bigger way. Explain this, please.
Yeah, I'm joined by my lovely young assistant here now. First off, I want to say that just like malaria, dengue is a complex disease that's, oh my gosh, she's waving now.
It's all right.
It's a complex disease that has many drivers, including, the drivers that govern where you see the vector and where people are exposed to bites from the vector and so on. But it also has fundamental thermal biology that we can measure using the trait-based approach that I described earlier. And what we find from this trait-based approach is that the optimal temperature for dengue transmission is much warmer than that of malaria. So dengue peaks at 29 degrees Celsius and malaria peaks at 25. We also know that the habitat for dengue vectors is different than Anopheles gambii. So we know that it is a container breeding vector that occurs in close association with people and often around urban and peri-urban areas. So looking at purely influence of temperature and temperature warming alone. Sorry.
Sorry.
We can say that the temperature is getting warmer and more suitable for dengue at the same time that urbanization and expansion of the Aedes vector is driving increased dengue risk. I'll let someone else finish commenting on that because I'm getting interrupted a little bit.
I can talk a little bit about the paper that you have on the left of the screen.
Please.
So in this paper, we essentially looked at, what the difference would be between the malaria curve and the dengue curve from our published papers, and some case data for febrile diseases in kids in East Africa. And there was this interesting pattern where some of the febrile diseases that were not classified as malaria fell into dengue temperatures. And so this sort of provoked this question of, could there be this shift? Might there be this shift towards dengue? And I think in follow-up work,
and
Desiree's team has done more dengue testing.
Sadie, just for clarification, the temperature we are talking about here is ambient temperature, not mosquito thermal limit, no?
Right. So this is, sort of ambient temperature at the study site. If I'm reading this one correctly, because I know there's been a few papers with these data in, and so this sort of led us to ask this question: would when we map out these sort of thermal boundaries, and then I projected them onto these climate models, do we sort of see a shift in where we see high density human populations and this seasonality increase? So more months of the year at those suitable temperatures for transmission. And we see this sort of shift across our scenarios where dengue can sort of just expand through Africa. And as we see those temperatures getting too hot for the current estimates of malaria in West Africa, we sort of see that dengue temperatures sort of prevail there. And so it's sort of it's a warning map. It's a tool for thinking about intervention in terms of, I always want to tie this back to how the decision makers might perceive it. For me, it sort of says, hey, let's start doing a bit more surveillance for dengue and dengue vectors in these places that we think there could be a shift, just to see if that really is happening, that can help explain these non-malarial febrile diseases, and to raise awareness that dengue is, quite prevalent in Africa once you start looking for it.
Yeah, one thing I want to add to that is this work was done in collaboration with my colleague Desiree Lebeau, who's the senior author here. She's spent about 15 years studying arboviruses in Africa, in Kenya specifically. And she's found in her studies that once you start testing for dengue and chikungunya, you see really high levels of infection, seroprevalence of 20% or more for dengue, for example, in the populations where she works in Kenya. So I think that the question of misdiagnosis came up in the chat earlier, and that happens a lot for dengue. So a lot of febrile illness gets misdiagnosed as malaria when it's really dengue, basically arguing that dengue is already a lot more widespread than is really recognized in sub-Saharan Africa, and that this problem could get worse as we kind of make that simple transition from temperatures that are optimal for malaria to temperatures that are optimal for dengue. Of course, acknowledging that there's a lot else going on here, including urbanization and including invasion of an urban malaria mosquito. But with all that in mind, oh my goodness, sorry. With all that in mind, dengue is already here in Africa, and it's a really big priority public health wise, starting to understand and control that burden much better.
And I think that, as I said, some of these mapping efforts, I think sort of are, for me, they speak a lot to the surveillance side, because that's our sort of first line of thinking about how to think about where we might need to intervene on the disease side. And so I think, this definitely hints at, and I think Erin and I have both sort of made it clear that there's a lot more going on than perhaps the focus of efforts have been directed towards so far at sort of, large policy grabbing scales. And so I think, I would say people need to look in these areas a bit more. And I know that people are now doing that a lot. And so I think this, as I keep saying, these are sort of the first approach efforts to draw attention to these.
Yeah, and again, this is a case where the impact is happening much within the populations rather than, at the ages. Like,
right.
There is a related question here.
All right, go ahead.
There is a related question here, related item here, which is with urbanization. So you have urbanization as well going on at the same time, and climate change, and both of these are risk factors significantly for dengue. In that, so how does that play out? Do they magnify each other? Do they synergize?
So
we did see some.
We think of climate as this sort of risk amplification in contexts where we already have urbanized landscapes. But when you also have urbanization as a process on the landscape, where maybe what we mean is we're converting sort of what we think of as natural habitat, which may not be natural, it might be agricultural habitat that's being taken over by some kind of built infrastructure, the human footprint on the landscape. I'm a geographer, so I'm always going to say we have to think about place and space. So what we call urban in some places looks very different from what we call urban in other places. But when we say urbanization, we're sort of thinking of this anthropogenic footprint expansion on the landscape and a built environment. And so that gives you different kinds of larval habitat. It gives you different types of occupational exposures. And obviously that then impacts how intervention can happen, because your road systems might be different, right? Like literally the distribution of bed nets versus distribution of spray teams is going to be impacted by what your infrastructure allows for. And so there's sort of multiple lenses of what's happening actually in these contexts. And we are shifting what kind of larval habitat availability there is. And at the same time, we might make bold assumptions about what kinds of things mosquitoes like, and then discover they do perfectly fine in other kinds of larval habitat as well. And so that's where this question of, when you say container breeding, what kind of container are you talking about? You know, is it a container,
and
or is it a puddle by other means?
And also just the heating effects that you have. We see this also with the urban ecosystems. There are some papers. You know, the housing structures are changing across the landscapes. And then in the city, you have also these heat islands.
Exactly. Where you have sort of more built environment, you are increasing sort of the acceleration of potentially shifting climate impacts as well by urban heat island effects, and by reduction of natural habitats and shade provided. But as I alluded to, I think this word urbanization can be complicated, because as you pointed out, there's many different housing types, and those different housing types also provide different resting habitat for mosquitoes and different interactions between people and where they get bitten. So
no, thank you very much.
Let other people take this one too.
No, we have five minutes, so I'm gonna push the last question, and then I'll ask the students to take the last shot. The last question on the humidity. This is a topic I really don't want us to miss. Where are we here? Yes, nobody talks about this. Everybody thinks climate change is temperature. And Dave mentioned a little bit earlier when he was explaining the impacts of climate change elements on malaria transmission. And this is a little bit complicated, but I think important to just highlight briefly. So I would like to start with you, Dave, and then Leah, and then we can take last comments from each of our speakers. What do you think should be done here, and what do you think are the impacts of humidity? We do have some studies already have indicated, for example, the Brown et al. Article does have some very nice interaction curves here, how temperature and humidity interact. We do have a very early demonstration from Nabi Bio's work, where they actually measured humidity after the rains, temperatures after the rains, and they could see significant changes there. Also from Nabi, the relationship between climate and relative humidity. I'm skipping this through this fast. And lastly, a recent study from India that indicates that including humidity actually boosts the predictive capabilities of these models. How do these things play in? How do we bring hydrology into all that? Dave, please, some explanation, and then what we need to do about it. Back to you.
Yeah, I noticed Leo was an author on one of the papers, so I'm going to be very brief and just say I've been convinced that by these papers that are looking at humidity and temperature together, so relative humidity as a measure, in part because they seem to work better when you're trying to explain the patterns of transmission. From a mechanistic point of view, though, I think it's really important to recognize that these thermal curves are, one part of the story. But if we think of mosquitoes as being adapted to an environment, and as desiccation as being one of the chief hazards that mosquitoes have to deal with, and I think it makes a lot of sense to think of humidity as affecting mosquitoes' decisions in as many ways as temperature does. And so I'm just going to leave it there, and I'm actually going to ask Leah to talk about this because she's a co-author on it, because I feel like I'm not as qualified.
Well, and I'll preface this by saying that too. I think that one of the reasons. So as Dave was saying that whether or not these mosquitoes are drying out and survive and are able to do all the things they need to do in order to be able to transmit relies heavily on humidity. And I think we're really trying to understand now better how temperature and humidity interact, because of course, if you think about a place that's hot and dry, those individuals are going to dry out more quickly than if it's hot and humid, for example. So there's definitely going to be an interaction. And I think that kind of understanding mechanistically what the impacts of this are is tricky. On the other hand, I think that we think that it's probably easier to understand kind of as a kind of as a general idea how humidity and temperature would interact to impact transmission than temperature and rainfall, for example, because as Dave was pointing out earlier, the impact of rainfall really depends on the local hydrology, right? So you have these very, very localized effects. So we're, you know, I often tend to be thinking kind of big picture, like what are the general things that might be going into where we see a lot versus a little bit of this transmission. And I think that that's kind of why we tend to focus on the humidity versus the rainfall. Although, again, in our individual studies, like when you look at a particular place, when the rain happens, has a huge impact on the number of mosquitoes that you see and the transmission that you get in those cases. So I think that part of the reason why this has not been addressed from this mechanistic perspective and has primarily been looked at through these association studies is because these experiments are very, very difficult, right? Where you're doing life history experiments. There's the paper from Nina from earlier looking at the phenotypic adaptation to try to understand adaptation to temperature.
Yeah.
Like we're talking 40,000 mosquitoes that were individually followed, right? It's a massive undertaking. So to be able to kind of describe mechanistically what's going on requires a huge amount of effort. And in mosquitoes, because it's so obvious and it's that of course temperature should impact it, and that's a relatively easy thing to look at, that has often been a focus. That's not the case for other vector-borne diseases, such as tick-borne diseases, for example, where humidity has been a primary focus and there's less temperature work, which is kind of an interesting difference between these different VBD systems.
Right.
So I think that, and interestingly, we think that humidity could impact more than just desiccation, right? So that, like in some of the preliminary work that Joel has done, who is the postdoc working with Courtney here, so he's the first author here, it's looking like you actually can still have shifts in many traits for humidity, and it changes the thermal response in very surprising ways that are not just about desiccation, which is really fascinating. So I agree that this is something that we really need to explore more of, and I think that we will start to see and be able to incorporate in this more mechanistic way, bringing in our laboratory data to try to understand and improve our forecasts in a way that doesn't exclusively rely on the correlational studies that we're already seeing some evidence for, but like we mentioned, can be difficult to pin down like the magnitude of those effects because of our issues with correlated predictors, for example. So I think that these are kind of nice complementary studies that are helping us to get a much better overall picture of what's going on, although that picture is still incomplete.
And on that note, I would like to say a big thank you to all of you. Before we close, I would like to request each one of you to provide your last comments. It could be on any of the topics, whatever you want to say. And this is also the same. I'll also request my colleagues from the University of Glasgow to provide their final comments. Each one person, just take a minute or 30 seconds to say your last words. It could be comments about the masterclass. It could be a response to scientific content. And we're going to do one last round in the remaining 2 minutes. Oh, his time is up actually in the next 2 minutes, so that we bring it to a close. But really, thank you all so much. I'm going to begin with you, Ivan, and then our other colleagues. I hope they're still in the room. Very quickly, each person, and then we go to our experts, and we will round it up that way. Start with you.
Hello, can you hear me through Najat's microphone? Yeah, no, I just wanted to thank you all really very, very much. I did have a few more questions, but they are on a slightly different topic, so I will not open up a new topic now. I'll just save them for later. But yeah, thank you very much for this incredible discussion, for making it accessible as well to younger or earlier career researchers. I'm just a second-year PhD student. I was, as every time I join these masterclasses, I'm not sure if I'm going to be able to follow up with everything. So thank you to all of you as well to make this accessible to people wanting to learn more and to get a bit more established in this field. And yeah, really looking forward for the following masterclasses. Also would like to apologize for the bit of chaotic of the beginning. This is the first time we're doing this. But yeah, thank you very much, everyone.
Thank you. Let's keep going. It's no time.
So the next, so I also would like to give thanks to our speaker. The discussion was really productive, and it was very vibrant, and we learned quite a lot. And special thanks to experts for being able to like respond to some of the questions. So I was trying to follow up what questions were not responded, but most of the questions were able to be they were receiving answers, and I'm very happy to participate in this masterclass. I think it has been a wonderful masterclass. Thank you.
Yeah, I think I'm also just gonna echo the words of my colleagues. So really thank you to the awesome moderation throughout the long 3 hours. And so also thanks so much for showing us the kids. It's probably the highlight for me. Really happy to see them.
All right.
I would also like to say thank you. As a disease ecologist whose work is primarily focused on how climate change will affect mosquito-borne diseases in Scotland here, I find it very important to understand these topics a bit further and to actually pose a few more questions that I can answer in my research. And if it's hopefully okay, I could message you, Leah, for a bit further more information on humidity, because this is currently something I'm very interested in including within my research. So thank you again, all of you today, and thank you as well, FREDROS.
It's most welcome. I'm not sure if we still have any other candidates from Glasgow. If not, then we can start with Dave.
Yeah, I want to thank FREDROS for moderating. I think it was well done, and for hosting the conference and setting it up in the first place. I just want to leave with one final thought. Mosquito-borne diseases, I think, are an equity issue. They really are diseases of poverty, and the people who suffer from them are also the people who are most likely to get affected by climate change in many other ways. Maybe we're putting the cart before the horse here. We should be looking at climate change, as malaria as a way of mediating the effects of climate change, not vice versa. So that's my final thought. Thanks.
Thank you, Dave. Thank you so much. And anybody who wants to go next?
I can go ahead and go. I kind of got to have a final word a moment ago, but I want to emphasize that I think that what you'll probably notice is that we have a lot of folks looking at this from many different perspectives, and having all of these perspectives, kind of like how Madeline mentioned that having different types of models improves forecasting. Having different types of perspectives, I think, improves our ability to understand what's going on. So I really appreciated having lots of these different perspectives here today. And I would also like to give a shout out to all of the graduate students that presented at the beginning of this. I know that it's kind of nerve-wracking to do that sort of thing in front of a lot of people you don't know, and people who are your seniors. And we appreciate you. And I think that I can probably speak for all of us who are the experts here, that we still have a lot of learning to do. And we're also keen to help you guys learn. So please feel free to reach out. Thank you.
Thank you
Thank you
so
so
much.
much.
I'll jump in next and just say thanks again to FREDROS and the team at Glasgow, echoing what Leah said, that I think you did a really, really nice job with that introductory stuff, and it is nerve-wracking. I'm nervous too, so it's a lot of people here. I also appreciate that we had multiple perspectives and were able to cover quite a lot about sort of the mechanistic modeling side, and then thinking about how that scales up and what that means when you scale up outwards and back down, for thinking about things like decision making, connecting into a lot of what Madeline was touching on. And so I think it's really important to sort of have these multiple perspectives in mind, especially for the grad students on the call, as you go forward, because there really are, all these issues do sort of end up in a place. And, you know, I ended up in a geography department, and I think that's great because context is everything. And so as we think about sort of spatial versus place, I really appreciate that this conversation today was able to touch on some of that and sort of be able to bring that to the forefront when we're thinking about what do we do with the information in the places we do things. So thank you again.
Thank you so much for looking.
I'd like to jump in and say thank you to everyone who participated in this panel, including FREDROS for organizing and the students who presented at the beginning and did a beautiful job. And also thank you for the fantastic preparation that you did with, you know, so many great slides that really covered so many aspects of the background. This has been one of the most interesting and unique and, you know, long-ranging conversations I've been involved in this topic. So thank you all. I'd like to leave with one last thought I didn't get to share, which is one of the big influences on my thinking in this area has been from Raina Plowright's work, who tends to think about the problem of zoonotic spillover, but it has a lot of overlap with vector-borne disease transmission, which is that, you know, we tend as humans to look for one simple cause for things, but actually when complex systems change and when events happen, they happen because of an alignment of many different factors that come into place. So when we've been talking about, you know, insecticide resistance and novel vectors and land use change and people's behavior and movement and drug resistance and mosquito evolution and thermal biology and humidity, all of this stuff comes together, not in a way that makes it intractable and impossible to understand, but, you know, when we're thinking about the impacts of climate, all those other factors have a role, and often all those other factors have to be in place in a way that's favorable for transmission in order for a change in climate to influence transmission. So I think capturing those multiple perspective is really valuable. And, you know, maybe we're all kind of touching the different parts of the same elephant here and thinking about how these changes are coming together to drive changes in malaria. And I really appreciate the context of what are our levers for control as well. So I'll stop there.
Okay, maybe I can come in now and just say a huge thank you for just inviting me to participate in it. It's been a big trip down memory lane, I have to say. It's just so nice to see colleagues that I've engaged with and read about and sort of interacted with over many, many years. I think it's obviously a lot of questions still to be answered, new questions that are emerging all the time. There's plenty more stuff to do. And it's just been a really exciting and interesting conversation. So thank you very much for involving me in this.
Thank you. Thank you so much for making it. Do we have Noor? I think he left. Anybody left? Did you finish? Students, experts? Going. Well, thank you all so much for making it till the end. This was a long one. We had 3 hours, nearly 400 people from the start, people coming in and leaving, and still now 100 people still with us on 3 hours. So I'm very, very grateful for all of you. I do hope that you will accept to join our next class if we request. You could notice that there's tons of stuff we didn't cover. But really just appreciating that you were able to spend 3 hours of your time with us. Thank you so much. We're going to bring this to a close, and we wish you all a wonderful night, a wonderful day, a wonderful weekend, and a happy Christmas, and see you again next time. Thank you so much. And thanks to my colleagues from Glasgow. Really, really amazing. Really grateful for your support.
Thank you.
Thank you all.
Thank you.
Thank you.
If you're there still, Fredros, well done.
Thank you. Bye.
Fredros, can you see my message in the chat? Might have gotten buried because there's a lot of chat going on.